Hello, check. Hello, hello, hello.
This call is now being recorded.
Hello, check. 1 2 3. Hello, hello, check. Hello, 1 2 3. Hello. Hello, check. Hello, 1 2 3. Hello, check. Hello. Hello, check. Hello, 1 2 3.
However, students, this third semester, sixth semester students, will join. Many of them have joined that. Okay. ওরা কেমন আছে জিজ্ঞাসা করতে হবে। How are you, my student?
Okay, Dr. Supratim Mandal is with us now.
Okay. Welcome.
Good evening, everyone. Welcome you all on today's webinar, Flaws of COVID-19, jointly organized by IQSC St. Paul's Cathedral Mission College and the Department of Microbiology, St. Paul's Cathedral Mission College. We have time for questions at the end of the session. Now, without further ado, I will
Thank you, Professor Daj.
Thank you, Peter. also very interesting. We are all in the cause of maybe COVID now. COVID-19. I will be with you all, and there's Dr. Supratim Mandal and you are so kind enough to access the invitation and two lines on the first word. And in this time, I think we will hear some news. But we are going to get our very also virus, but it has a vector. This one has two vectors. Therefore, naturally, with a virus, again, the problem is here that uh, is that there is some good unique mechanism which is the heart, which is really not in RNA virus. It has some and I have and thank you.
Thank you, sir, for your motivating words. We are also privileged to have with us today Professor Supratim Mandal, the coordinator of IQAC, St. Paul's Cathedral Mission College, who will grace this occasion with his encouraging words. Can we please have Professor Supratim Mandal? Do we have Professor Supratim Mandal with us?
Yeah.
You with us? Professor Mandal?
I think Sal has some network problems, so we can continue for our first session.
In that case, we would now continue with our scientific session. Am I audible to everybody?
Yes, you are audible. Yes.
Okay. Professor of Microbiology, University of Kalyani. Dr. Mandal has done his PhD work in microbiology, experimental parasitology, to name a few. He is also a member of prestigious organizations like American Society of Investigative Pathology, European Society of Medical Oncology, and Indian Science Congress Association. Now, I would like to request Dr. Mandal to carry on with this scientific session and enlighten all of us with his valuable research ideas. Over to you, Dr. Mandal.
Good evening, everybody. Am I audible?
Yes, sir. Yes, sir.
Okay, thank you for the generous introduction. First of all, I would like to show my gratitude to thank the Honorable Teacher in Charge, Professor Adhiraj Roy, St. Paul's Cathedral Mission College, Kolkata,
The IQAC coordinator, Professor Shobhajati Mohapatra, Bursar, Professor Shubhendu Kanjilal, and Professor Abhishek Dash and other faculty members of the Department of Microbiology. It also gives me immense pleasure to share the platform with my friend Dr. Adhiraj Roy from Eminent. So I'll be starting my presentation now. It's not coming. Just give me a minute. Okay, you can see the slide. So the title topic, COVID-19 from origin to search of an antidote. Now, in recent decades, there are several new diseases that have emerged in different geographical areas, with pathogens including Ebola virus, Zika virus, Nipah virus, and coronaviruses. Now, recently, as we know, a new type of viral infection emerged in Wuhan city, China. And initial genomic sequencing data of this virus do not match with previously sequenced coronaviruses, suggesting a new coronavirus strain which has now been termed as severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. Now, in today's presentation, I will try to address the epidemiological, clinical, and pathological aspects and the probable therapeutic strategies that have already been globally recommended to counter this pandemic virus. Now, coming to a brief history of the pandemic, the Nobel on 12 December 2009. Now, on 11th February 2020, the World Health Organization or WHO announced the official designation for the current coronavirus associated disease to be COVID-19, caused by SARS-CoV-2. But the primary cluster of patients was found to be connected with the Wuhan, South China seafood market in Wuhan. The COVID-19 emerged in China and spread rapidly throughout the country, as we know. Now, due to the severity of this outbreak and the potential of spreading on an international scale, now initially the epicenter Europe, particularly and now the United States, India, and 5 million infections. Now, first I would like to address the structure and what we know about the structure of the virus. Now, SARS-CoV-2 is a member of the order Nidoviride, family Coronaviride, and subfamily Orthocoronaviride, which is subdivided into 4 genera: the alpha coronavirus, beta coronavirus, the gamma coronavirus, and the delta coronavirus. The genera alpha coronavirus and the beta coronavirus, they originate from bats, while gamma coronavirus and delta coronavirus, they are evolved from bird and the pig genus. Virus order, the Nido Latin being nested. Virus order viruses, they depend on a nested group of mRNA for their replication. They contain the biggest known viral genome found made, sizing up to 27 to 32 kilobases. Now, these viruses are enveloped positive sense single-stranded RNA viruses. Now, under electron microscope, the virus shows the characteristic crown-like shape due to the presence of the spike protein on their surface. Now, the spike protein is extensively glycosylated, forming a homotribe. The ectodomains, that is the outside domains of all coronavirus yeast proteins, they have similar domain organization. Game that has a major role in viral assay. And the nucleocapsid protein, that is N, it is responsible for the regulation of the viral RNA synthesis. It interacts with the M protein during the budding of the virus and forms part of the nucleocapsid in association with the viral RNA. The SARS-CoV-2 has sequence homology with the influenza virus through its hemagglutinin esterase glycoprotein known as HE. Now this HE also it binds to neuraminic acid on the host cell surface, leading to the adsorption of the virus on the cell surface. Now, within the first incident, the Chinese scientists, within 1 month, they have successfully sequenced the whole genome. And when the SARS-CoV isolates analyzed phylogenetically with related coronaviruses from past human outbreaks of animal origin, including Mars coronavirus, bovine coronavirus, canine coronavirus, bat coronavirus, bat SARS-like coronavirus, and equine, that is the horse coronavirus, along with all 5 defined subgenera of the beta coronavirus, namely sarbecovirus, embecovirus, marbecovirus, nobecovirus, and hebecovirus. So the results show that the nearest neighbor of SARS-CoV-2 were the bat SARS-like coronaviruses. Furthermore, the nucleotide present in virus sequences, the known with the SARS-CoV-2 isotypes and bat SARS-like coronaviruses, indicating common origin. Now, the potential transmission routes for SARS-CoV. The possible origin of SARS-CoV-2 and the fast mode of disease transmission are not yet clearly identified. But the analysis of the initial cluster of infections suggests that the infected individuals had a common exposure point, which is a seafood market in Wuhan, China. Now, the restaurants of this market, as well as they are known for providing different types of wild animals for human consumption. As you know, the Chinese, they eat a lot and of type of exotic animals. Now, the Wuhan South China seafood market, they also sell live animals such as poultry, bats, snakes, and marmots. So this might be the point where zoonotic, that is animal to human transmission occurs. Although SARS-CoV-2 is alleged to have originated from an animal host with further human-to-human transmission, the likelihood of foodborne transmission should be ruled out, since it is a latent possibility. Additionally, other potential and expected routes would be associated with transmission, as in case of other respiratory viruses, by direct person-to-person contact or by contact with contaminated surfaces. Still, and organ transplantation, as well as translational and perinatal routes, that is from for SARS-CoV transmission of determined. Now, from experience with several outbreaks associated with known emerging viruses, the higher pathogenicity of a virus is often associated compared to emerging viruses, Ebola, avian flu, and MERS. SARS-CoV-2 has relatively lower pathogenicity and moderate transmission. Now, following the initial infection, the human-to-human transmission has been observed with the preliminary reproduction number, that is R0, which is an important number, that was initially estimated as 2.24, and now it was found to be 3.58. Now, this is the problem. The higher R0 value is indicative of the more significant potential of SARS-CoV transmission in a susceptible population. So the virus is evolving, it is mutating, and as a result, the R0 is getting higher, and the potential for transmission in a susceptible population, that is the disease transmissibility, is getting higher. Now, next I want to give a brief idea about how this virus, what is the life cycle of this virus, how this virus enters the cell, and what is its replication strategy. Now, it has been confirmed that SARS-CoV-2 viruses They use the same cellular entry receptor as the other SARS-CoV. So the receptor name is ACE that is found in the lower respiratory tract of human. The virion S glycoprotein that is present on the surface of the coronavirus, it attaches to the receptor. This leads to the cleavage of the trimeric S protein, and that is initiated by another cell surface associated serine protease known as PMPRSS2 and another protein that is known as cathepsin. Now this S glycoprotein includes two subunits, S1, another one is S1 is responsible for the determination of the virus host range, and while facilitates virus cell membrane fusion by two tandem domains, the heptad repeat 1 known as HR1 and the heptad repeat 2 known as HR2. Now the DNA of coronavirus are approximately 30,000 nucleotides long and encodes for structural proteins and non-structural proteins of the virus that have a critical role in viral RNA synthesis. So at least one niche-specific protein, that is non-structural protein 2, known as NSP2, and one structural protein, that is the nucleocapsid protein N, they are involved in viral RNA synthesis. Now the expression of the coronavirus replicase transcriptase protein genes is mediated by the translation of the genomic RNA. Now the replicase transcriptase proteins are encoded in two open reading frames. One is the ORF1A, another one is the ORF1B, and they are synthesized initially as two large polyproteins, that is PP1A and PP1AB. Now during or after the synthesis, this is split by viral PL into 16 small proteins. Now the replicase transcriptase proteins together with other viral proteins and possibly some cellular proteins assemble into membrane-bound replication transcription complexes known as RCT. Now these RCT complexes accumulate at perinuclear regions and are associated with double membrane vesicles here. So finally the virion containing the vesicles, it fuses with the plasma membrane of the cell to release the virus, and the virus then attaches a new cell and the replication is repeated. So now coming to the pathogenicity of the virus to the respiratory tract, the cilia in the lower respiratory tract, it facilitates the attachment of the virus to its receptor. The multiple SARS-CoV-2 proteins interact with components of the innate immune system to evade an antiviral interferon response. The main pathogenesis of COVID-19 infection as a respiratory system targeting virus is severe pneumonia combined with the incidence of acute cardiac injury. Now, acute respiratory distress syndrome, known as ARDS, is the main cause of death in COVID-19 disease so far. One of the main features of cytokines by immune effector cells. Now, cytokines are basically a large group of proteins secreted by cells of the immune system that acts as signaling molecules that mediate and regulate immunity, inflammation, and hematopoiesis. Granulocyte, stimulating factor, interferon gamma, MCP1, IgG, T1 necrosis factor alpha. Cytokines response that contributes to ARDS, multiple organ failure, and finally death in severe cases of SARS-CoV-2 infection, similar to SARS-CoV and SARS-CoV infections. The patients infected with COVID-19 showed higher leukocyte numbers, abnormal respiratory findings, and increased level of death from acute COVID-19 patients, involving cytokine storm damage to lungs and multiple organs of the body like heart, kidney, and liver, leading to the multiple organ exhaustion. This is the pathobiology of the COVID-19. Now next, coming to the symptoms. The symptoms of COVID-19 infection appear after an incubation period of approximately 5 days. The period from the onset of COVID-19 symptoms to death, it ranged from around 6 to 41 days with a median value of 14 days. Now this period is dependent on the age of the patient and status of the patient's immune system that you have heard about the comorbidity, but I'm not going to deep into that because the other speaker, Dr. Adhiraj Roy, will cover that area, I think. So it was shorter among patients greater than 70 years old compared with those under the age of 70. Now, the most common symptoms at onset of COVID headache, diarrhea, and phase 3, severe potentially lethal disease with hypoxia, ground glass infiltrates in the lung, and progression to acute respiratory distress syndrome, that is ARDS, with very high viral load. Now, the question comes into the mind is that why there is a delay in the availability of a drug or a vaccine. You are saying why the vaccine is not coming. As student of biology, we know the problem. But the basic thing is that we need to first know and understand the critical route for a ration. Biological process under studies and ration. So then comes the safety and efficacy of the most promising candidates, which would be then initially examined in various animal models to ensure that these findings also will work in humans. But something is working on a mice, not definitely work on a human unless you go step by step and test it on a higher mammal also like monkey. So next, the few lead candidates would then be screened out and trial through three distinct phases. This is very important. Phase 1, and the review of the drug end. Now, fulfilling all the previous criteria, an expert regulatory body then scrutinizes clinical trial data and decides on approving a promising therapeutics. Now, this process is a typical 15-year long process. So that we expedite the typical 15-year track of a therapeutic from discovery through its commercial availability. A COVID-19 accelerated drug development initiative has been announced to shorten this process from 15 years to 18 months. And now the company, they are working day in and out to make it much sooner than that. So you can imagine the kind of work that they're going through to get a drug candidate or vaccine. Now, how to design a potential drug target for this pandemic? As we know, we now know the structure of the virus, the pathogenesis mechanism, design the potential drug targets. Now, previous coronavirus pandemics like MERS and SARS, the considerable gone into the search for drugs and subsequent drug candidates. Now, based on these and life cycle stages of SARS-CoV virus, the therapies that have the potential to act on coronaviruses can be divided into 5 approaches. Okay, so first approach should be inhibition of virus binding to the host receptor by either chemical compounds or monoclonal antibodies. So these agents will block or effectively engage the host cell surface receptor, thereby preventing the entry of the virus and subsequent internalization. Now, second approach should be let them enter the surface but target cytosis. This process enables us to enter the virus cell and release its material for further replication, and therefore blocking virus-mediated endocytosis is a logical target for antiviral therapy. But the third approach should be to neutralize the virus particle itself. So this can be accomplished by the compounds and antibodies acting on enzymes or functional proteins critical to viral replication multiplication. So the fourth approach will be the targeting the viral structural proteins like the membrane, envelope, and nucleocapsid protein, thereby blocking the virus repackaging. Okay, to stop the new virus particles to generate. And finally, restoration of host innate immunity by the agents capable of producing virulent factor against this pandemic. Now, the first drug that we have heard about is the chloroquine phosphate and hydroxychloroquine. Now, chloroquine is an old drug for treating malaria. Now, it increases the late endosomal and lysosomal pH, leading to impaired release of the virus from the endosome or lysosome. Therefore, what happens is the virus cannot replicate because it is unable to release it generated in the cell. So these drugs seem to act as a that is the interesting part, which permits the chloroquine phosphate. So this is the reason. They are assuming that the gene will act as zinc ionophore and boost up the activity of the drug. Now, since it increases the endosomal pH, which is required for viral cell fusion, so it has a potential broad spectrum viral activity. In addition, it is also found to be interfering with the glycosylation of cellular receptors of SARS-CoV. Now, another member of the same chemical family, that is the hydroxychloroquine, is also in the news. So it is on plasmacytoid dendritic cells. Now, these toll-like receptors, known as TLR, especially TLR, recognizes DNA containing complexes, result cells the TLR help to support the system so that there should not be any cytokine storm. 800 patients in the University of Minnesota Medical School, they found out that it does not treat the COVID-19 infection. They published this data. So in June, in the UK, the use of hydroxychloroquine was discontinued. When an interim analysis showed it provided no mortality benefit to people hospitalized with severe COVID-19 infection. Okay. On the contrary, a randomized trial of patients with mild symptoms in Brazil. Now, since a good deal of inflammatory response it is observed in COVID-19, so any good anti-inflammatory drug, especially those that block the major inflammation pathway, that is the JAK-STAT pathway, they can be used also for COVID-19. So one drug that is quite useful and well known to treat the rheumatoid arthritis, the high affinity kinase bind like remdesivir increases the potential of the remdesivir to lessen the infection. So hence it could be trialed using an appropriate patient population with COVID-19 to reduce both the viral entry and also the inflammation in patient. And it is now in fast phase trial. The next another interesting drug that I am going to tell is the remdesivir. Now this is sold under the brand name of Eclari. It's a broad spectrum antiviral medication developed by GDR Sciences, and it was initially they developed it for treating Ebola. Okay, so although it was subsequently observed to show antiviral activity against but instead is late. For the RNA-dependent RNA polymerase of SARS-CoV or SARS-CoV-1 or SARS-CoV-2, the arrest of RNA synthesis occurs after incorporation of 3 additional nucleotides. Now, As a result, remdesivir is classified as a direct-acting antiviral agent that works as a direct terminator. The trial phase 2 trial is currently going on in USA with this drug. Now the next drug, that is favipiravir, it is a pyrazine carboxamide derivative which acts against many RNA viruses. It is also marketed as T705, Avigan, or Favipira in different names. It is very closely associated with another antiviral drug, that is ibavirin. Favipiravir, it functions as a prodrug. So initially the compound is not active. When it undergoes a change inside the cell, then it will become active. That's a prodrug. So the favipiravir functions as a prodrug and undergoes ribosylation and phosphorylation intracellularly, become the active favipiravir RTP. Now this active favipiravir RTP selectively inhibits the RNA polymerase and prevents replication of the viral genome. Although this drug was originally developed to treat influenza, this RTRP catalytic domain of the RNA polymerase, which is the drug's primary target, is expected to be similar for other RNA viruses also. So this conserved RTRP catalytic domain contributes to favipiravir's broad spectrum coverage. Now, from the phase 1 trials of favipiravir in US, patients showed that favipiravir had more powerful antiviral activity than that of the lopinavir or ritonavir. Now, another drug that is basically developed as an HIV drug is known as Kaletra. It's a combination of lopinavir along with a little amount of ritonavir. Now, it is usually recommended to be used with other antiretrovirals. The lopinavir is an inhibitor of the HIV protease that is made by the Abbott Laboratories, which is a US-based pharmaceutical company. Now, lopinavir inhibits the HIV protease compound by developing an inhibitor enzyme complex, thereby inhibiting the cleavage formation of the group-specific enzyme antigen polyprotein in HIV. HIV proteins. Now, these protease cleaves protein precursors and develops new viral particles. Now, protease inhibitors interrupt this cleavage process and interrupt the formation of new viral particles. Now, initial results from the clinical test comparing a lopinavir and the ritonavir combination, the quality of care of COVID-19 suggests this therapy is also safe. Now, I am coming to another therapy which is known as the convalescent plasma that you have heard of maybe in the news. So there is a blood plasma therapy. So it is not using the medication, it is using the OC system when one person used to treat another person. So basically what is this? This is the yellowish liquid component of the blood that is consisting about 55% of the body's total weight, total blood volume, is the blood plasma. Now this fluid part of the blood conveys cells and proteins all through the body. Now, when the body is attacked by a pathogen, our immune system produces antibodies which help to fight off the infection. But if the infected person produces enough antibodies, he can recover from the disease caused by that pathogen. This is what is happening in the asymptomatic patients. So the idea of blood plasma treatment is that such immunity can be transferred from one healthy individual to a contaminated one utilizing the blood plasma. Now, in this method, blood is drawn from someone who has recovered from COVID-19. And then the serum is isolated from the volunteer and screened for antibodies that can neutralize the virus. So the serum which is rich in antibodies is then administered to another COVID-19 patient having the severe symptom. Now, there are certain limitations of the process. That is, you need to have a huge number of volunteers. Now, there should be a variability from batch to batch. The antibody titer might be different. And also one important different form of this particular method. So what they are using the monoclonal antibody as a neutralizer. So the monoclonal antibody administration is an alternative to the convalescent plasma therapy. So there might be two potential mechanisms of coronavirus antibody neutralization. The first one is neutralizing antibodies could block the viral infection by binding to the viral spike protein and preventing it from interacting with the cellular receptor, that is AC2. should be the neutralizing antibodies could bind to the viral spike protein and block the conformational changes that the spike protein must undergo to facilitate fusion of the viral and the host cell membranes. Now, another aspect is that antibodies also could enhance the viral entry into the immune cells by binding to the viral spike protein with their FFP portion and to the Fc receptors with their Fc domains. Now, among the drugs that I have talked about here, in all the cases, the patient needs to be infected and the drugs then will treat them. But to combat a pandemic, you need to create the immune memory so that your body should be prepared before the pathogen enters your body. So that is why generation of the vaccine is the most effective way. So now coming to the vaccine for COVID-19. So first of all, the vaccine should be very easy to administer. An oral or international national vaccine should is required. Is the human population of the world, and that process must be feasible and rapid. And the third one is that long-term storage of the vaccine at room temperature should be a sought after goal. So the vaccine should tolerate the temperatures. To facilitate transport and stockpiling in especially underdeveloped nations with inadequate supply chain and cold room facilities. Now there are 7 main platforms of vaccine development that are currently being exploited by the companies. So there are 2 nucleic acid platforms. One is the DNA and another one is the RNA. The third category termed as protein-based, also known as the based vaccine. And represents the largest virus themselves. So in the case of the viral based vector, there will be 2 types. One is non-replicating and another one is the replicating viral vectors. So you will take one virus that is similar to the coronavirus, but it is an inactive form and it is not, and they change its genome, place coronavirus gene inside the genome and then use it as the vector. Okay. So, and the other 2 category, they are the SARS-CoV-2 virus itself. So, SARS-CoV-2 virus is used by itself. So it is either inactivated, usually with a chemical such as beta-propionolactone, which chemically inactivates enveloped viruses and can inhibit viral membrane fusion in a dose-dependent manner, or it should be a live attenuated virus that is generated by techniques such as serial passaging in cell culture. If you grow the cells in the laboratory one after another passage, the infectivity will decrease. The virus will be live, but there should not be infectivity. So that can be used as a vaccine. And you can see here another category is there, that is the other category. Okay. So the other category includes virus-like particles, the use of non-SARS-CoV-2 virus carriers such as killed rabies virus or live modified horsepox virus. So according to these criteria, you can see here there are over 125 SARS-CoV vaccines that are currently under development, which is astounding, from all across the globe in such a short time frame. So 125 vaccines are currently they are under trial. Now coming to the global race for vaccines targeting SARS-CoV-2. So these are the few vaccines that are leading the race. So I'll be talking to the shorter of time. I'll be talking only about the 5 that are currently in phase 2 or phase 3 trial. So they will the okay, this one. So this is developed by in collaboration of University of Oxford and AstraZeneca that is in UK. So here the antigen like 2x1. Now this is a weakened version of a common cold adenovirus with the spike encoding region that is cloned into its locus. Although it infects the primate, but it is genetically altered in a way that it is incapable of viral spread in humans. The same vector modality is in vaccine candidates for influenza, tuberculosis, chikungunya, and Zika virus. Now, what is the safety of this platform, of this vaccine? The vaccines made from the cheap adenoviral vector platform for over 10 different pathogens have been tested in thousands of volunteers to date and are safe and well tolerated. And although they can cause some temporary side effects, like these vaccines, the report was there that there's some temperature elevation, headache, or some sore arm where it is injected. But the advantages of this vaccine is that it is a favorable safety for efficacy, safety and immunogenicity in 10,000 volunteers in Brazil, UK, in our country, India, and South Africa. Their target is to produce this at the end of 2020. To the next one, that is from biological company named Moderna in USA. So here the antigen is the full-length pre-fusion stabilized SARS-CoV-2 spike glycoprotein. Now about the platform, it's a non-replicating RNA genetic material mixed with lipid nanoparticle formulation. Now about the safety, there are around more than 1,500 injections with Moderna's RNA formulations for Zika and chikungunya viruses. They have taken place in other phase 1 and phase 2 clinical trials previous and are generally well tolerated. But the problem with this one is that till date, no RNA vaccines are currently licensed and approved for use in humans for any disease. So this will be the pioneering one. So they are very cautious about it. At the end of the year, and they are getting huge fund from the US government for doing their work. Now coming to the third one, that is the BNT 162. It is developed by the collaboration of three pharma giants. One is the BioNTech AC, that is from Germany, the Pfizer from USA, and Fosun Pharma, that is from China. Now here the antigen is the SARS-CoV-2 TC-like protease, the nucleocapsid protease, that is NSP5, and another main protease, that is SARS-CoV-2 main protease. Now here the platform details is that hybrid containing mRNA or nucleoside modified mRNA that is mixed with lipid formulation target. So the advantage is also that the fast manufacturing trial like 30,000 individuals in US, Germany, Argentina, and Brazil. The target is to produce 1 million doses at the end of the year and 100 million doses at the end of 2021. Now coming for the fourth one, that is the AD5N-CoV, that is produced by the Cisco Biologics in China. It's a full-length SARS-CoV-2 spike glycoprotein. Now the platform details are the non-replicating adenovirus that is maintained in cells as an epitope. The essential genes are then deleted and replaced with a high activity cytomegalovirus, that is CMV promoter, which drives expression of the target base protein. The safety of the platform in general is safe and well tolerated. However, some reports are there that it can be dangerous for the immunocompromised individuals, and there is a problem of large-scale manufacturing. But interestingly, this is the only vaccine till date that is approved. It is approved by the government. So the Chinese military, they approved the vaccine on June 25th for 1 year as a specially needed drug for their soldiers. But till date, CanSino would not say whether the vaccination would be mandatory or optional for the soldiers. We need to wait for this. Actually, the current status outside China is that the company is undergoing the phase 2 trial for dosing and side effects. 5,000 it is. Now, finally, the CoronaVac, the fifth candidate, that is also known as the Ecovac. It is produced by the Sinovac Biotech China. But this is the only virus, the only vaccine that is using the whole virus. Okay. So the antigen is the whole coronavirus, with initial reports that the receptor binding domain, that is the RBD, using the spike protein is the main immunogen. The detail of the platform is that SARS-CoV-2 virus was plaque purified and passaged several times in Verocell to decrease the infectivity. Then it was inactivated with treatment with beta-propionolactone, followed by mixing with an adjuvant using alarm. The safety of the platform is it is the most safest among all because the vaccines made from inactivated viruses, they are used throughout the world with generally excellent safety profile. So it's a straightforward process of using inactivated virus, which is favorable safety and tolerability profile. It is currently the phase 3 trial is going on 1,200 individuals in China. So finally, I like to conclude by saying that applying quick fix and shortcuts can lead to errors and disastrous consequences. What regulators have to worry about is the atmosphere of hype about this vaccine. All data which form the basis of any findings need to be scrutinized and should be confirmed by other investigators. Relaxation on regulatory principles based on political pressure and goodwill needs to be resisted, and one needs to protect the interests of the volunteers who are a part of such experiments. So finally, I'd like to conclude with a positive note, with a quote from one famous American science fiction novelist, Robert A. Heinlein, that everything is theoretically impossible until it's done, and hope that we should be able to come out with these difficult times with the help of a vaccine candidate available in the market at the end of the year. I'd like to finally thank my PhD student, Mr. Shomik Bishash, who helped me in the preparation of the slides, and also thanking the organizers for giving me the opportunity to talk in this class. Thank you.
Thank you, Dr. Mandal, for the wonderful informative talk you shared with us. Now I would request Kumar Singh Roy, Faculty Microbiology Department.
The first question to you, Dr. Mandal, is from Shubhi Shah. He asks you, there is a rumor in the market, and according to some, they suggest antibiotics stated that under mild to moderate cases, such drugs are effective. So over to you, Dr. Mandal.
Doctors are saying it's just a supportive related to this, and there is no such intent of conclude that this really actually helps to prevent the COVID-19 infection or not. It's just a supportive drug.
Okay. We have a student, Omlan Banerjee. He also has a question for you. He says, many of these viruses, they come from bats, but bats are immune to them. Now, Omlan's question to you is, can we learn something from bats that would help us fight the virus in humans?
Yes, we can. Actually, the are not value I talked about that the thing is that the virus always that those viruses will try only that are not pathogenic. There is a confusion and panic among the people. The thing is that here before transmission we should not worry about that much. There is too much panic about it, but we should and we should see how what it is, what its life cycle is inside their vector. That's the interesting point. But no paper as of yet has come on how it actually modifies itself inside the vector. Something to think about later.
Ravi. He says, if hydroxychloroquine minimizes the cytokine storm, which is a potential symptom of SARS-CoV, then why is it used in the first place?
No, hydroxychloroquine, it is not a drug, it is not a drug that kills the virus. So it is the drug that so there is somehow a decrease or inhibition of the viral assembly. That's one part of it. Another part of it that it blocks the TLR pathway so that there should not be the activation of the cytokine production. So as the more cytokines will be produced, there should be generation of the cytokine storm, which leads to the lung injury, and it will be detrimental for the patient. So the disease at one side it is decreasing the disease pathogenesis, as well as in other side it is trying to kill the virus, which is not clearly understood yet. But
Okay. Hello, am I audible?
yeah.
Yeah. Okay. Just the last question to you, sir, is from Kagya Barua. She asks you, a person who has already been recovered from COVID-19 and can be infected again. Okay. She asks you that a person who has already been recovered from COVID-19 can be infected again. So in such a case, how can we assure that the plasma will be a successful strategy for the recovery for other patients?
So actually, till date, there is no such evidence. But thank God for that. That there is a recurrence. With the virus can reinfected with the virus also with the short period lifelong. But if we can say that immunity can be, we can say this after the patient need to the drop the blood type matching. So it is much more preferable now right now is that identify the monoclonal antibodies.
Yes. Okay. Thank you so much, Dr. Mondol, for lending us with such wonderful insights. Thank you so much. Now, we shall move on to the second scientific session, and it is our privilege to have Dr. Adhiraj Roy with us. Dr. Roy has completed his PhD from the George Washington University, United States of America in biochemistry and molecular genetics. He was a postdoctoral scientist in the Department of Biochemistry and Molecular Medicine in the George Washington University, USA. He later worked as a scientist in TCG Life Sciences, India. Dr. Roy is currently associated with Amity Institute of Research Specialization in the fields of biochemistry, signal transduction, molecular oncology, and molecular biology. He has published his research works in the domains of molecular oncology,
Dr. Supratim Mandal.
Yeah.
Can I share your presentation as a lot of students are asking for it?
Okay.
Okay, thank you.
Okay. So we shall move on. Now, I know that the list of the accomplishments of Dr. Adhiraj Roy is endless, and it is an absolute honor to have Dr. Roy with us. Meanwhile, as we progress through the talk of Dr. Roy, I would request the inbox to be addressed by Dr. Roy at the end of the session. The house is all yours. Dr. Adhiraj Roy, over to you.
Thank you for that wonderful introduction. I would like to express my sincere gratitude to the teacher in charge, Dr. Guru, and my friend, Professor Bash, to invite me
is present in the gallery. My voice is clear right now? Hello? Okay, now is it clear, everyone?
Yes.
Okay. So what I'm going to show you today, one of the major problems during this COVID-19 pandemic, and that is basically the issue with comorbidity. So you have definitely seen, you have definitely heard that there are a lot of fatalities going on due to not only the infective, so the comorbid problems the patients have. Diabetes, that may be and most importantly, because I am a cancer biologist, I am a little bit more concerned about the cancer patients. So today I am going to talk about the implications and the promise we have on the cancer patients during this pandemic. Let me share this slide. My talk today is COVID-19. Current scenario on the cancer patients. So how they are living in the pandemic. Okay? I will give you a very some seminal papers which scientists all over the globe published connecting COVID-19 and cancer. Okay. So we all know this famous pandemic, COVID-19, is actually caused by severe acute respiratory syndrome coronavirus 2. That is SARS-CoV-2, also known as 2019 novel coronavirus, 2018 influenza. So because this is a novel virus, the genome sequencing has been done on this virus, and it has a lot of sequence similarity with its predecessors, but it's a novel virus. That is why scientists really named it, gave the nomenclature as 2019 info. The transmission electron virus, you will see clearly there is corona means ground. Coronavirus. And this image was actually one of the first images that emerged in the scientific literature, and this viral particle was actually isolated from the patients in US. And this is a transmission electron microscopy image. So next to this, I will tell you that this is a false color imaging, because when you do transmission electron microscopy, you kill the virus or you kill any specimen, and then using software you can color them so that you can distinguish between different cellular components. Okay. So you can see here the green dots all over the at the surface of the virus. This is basically the spike protein. So the name derived from the spike protein. That's why it's coronavirus. And I'm just going to touch some of the salient so Dr. Terry was one of the pioneers in the foundation. So he actually discovered adenovirus belonging to the lentiviral group, and he actually isolated the virus from avian strain. So that was one virus. So basically it was avian mycoblastosis virus. So Dr. Roy, our candidate, SARS-CoV-2, it belongs to particle class 4, and it's a positive sense single-stranded virus. So this viral genome can act as mRNA. So it will self-act as mRNA, and it can code for its necessary proteins once it gets is inside a process. And some of the other class 4 viruses, those are kind of siblings to SARS-CoV-2, like MERS, Dengue virus, C virus. Do not even transmitting human from non-human, but in the market, it actually started transmitting. That is. Like Dr. Supratim already discussed a lot in a very detail about the structure of the SARS-CoV-2. So I'm not going to too much detail about it. So we all know. So this virus, these virion particles, viral particles of the virions, they are about like 70 to 200 nanometer in diameter, and that makes the virus very much vulnerable to fly through the air in aerosols. That is this 6 feet of distance. Like separate distance. And there are 4 structural proteins. The spike protein is responsible for virion binding to our host cell. The envelope, that is the E protein. There is a membrane from the M protein, and there is nucleocapsid. N protein. So nucleocapsid is very important because it acts as the scaffold of biology. So scaffold is the hold. So there should be a protein scaffold that should hold the biology. Nucleic acid, in that sense, is very significant in the biology itself. So when the pandemic actually started to hold the grip, this paper from SEM was one of the seminal publications. So once it all, paper is structure, function, and efficiency of the SARS-CoV-2 spike protein. So this seminal paper actually showed how SARS-CoV-2 interacts with the host cell. And in this paper, they elaborately describe the biology of disease, angiotensin EC2, that is the receptor expressing on the host cell. And this paper showed the interaction, biological interaction, molecular mechanism between the spike protein and so kinase from the paper came out as SARS-CoV-2 uses ACE2 to enter the organism. So they utilize ACE2 as angiotensin converting molecule to enter the host cell. So SARS-CoV-2 and SARS-CoV bind with similar affinities to ACE2. This is very important point because suppose some individual is already infected with SARS-CoV-2 and somehow we can imagine a hypothetical situation where there is a pandemic of SARS-CoV, then there is a very high chance the same individual will get both infection and spread. Because it is mostly expressed. So I just wanted to show you how ACT cells, one of the cells, they are all non-molecular. And the green is kind of expressed. So what you do, you take an antibody, it's that primary antibody, it is secondary antibody, antibody will fix the cells. Normally, or mentioned, these are uneven surfaces that gives you different properties. That's the whole surface in different properties. And then they scan all the properties. If you blow up the image, you magnify the and surrounding the airway, this is the basic question. So these are the some papers, like respiratory medicine, 2022, very current. European Journal of Internal Medicine, 2022. There is Journal of Immunology, that's a review. All these papers, they are all talking about the interaction between BCG2 and SARS-CoV. So they aim in BCG as a novel, as a new target, 0 from 1. Okay. So they all talk about how BCG2 interacts with SARS-CoV. As the molecular mechanism, can we use BCG2 as a novel therapeutic target to eliminate SARS-CoV-2 entry? So these are the different papers you can definitely go into part. This cytokine and chemokine storm very intense association. A very interesting paper, a very prestigious journal, Lancet, it came out and it actually showed that it is the first sentence that is very crucial for disease management. Not only the cytokine storm. So we all cytokine storm is it our system has been infected with a virus, immune system is activated, and we have a lot of cytokines and chemokines like interleukin, human necrosis factor, granulocyte colony stimulating factor, MCSA. So a lot of cytokine and viral sepsis. So viral sepsis is actually characterized by excessive macrophage infiltration. Macrophage is one of the living germ, one of the living soldiers. So when SARS-CoV infects the lung cell, there is an excess lot of protein coagulation going on in the lung cells. And increased fibrin degradation. So fibrin is a scaffold protein. It gets degraded. All these degraded proteins, they all get coagulated around the process. So this is called viral sepsis. And this viral sepsis, coupled with cytokine storm, it actually gets worse when people have cancer. So that is the sad part. So here comes the comorbid factor. So cancer patients, they have to be really, really careful. So I will just go through a very basic of cancer. You know, cancer and this group. This group it all starts with how cancer came to the play. You know, like he just put the latest figure. Like recently, there has been a discovery that Centrosaurus dinosaur they contracted cancer. So you can imagine if almost 70 to 80 million years ago, cancer was there. So it's a very old disease, it's civilization. So recently, this Canadian group they found the fossil of this dinosaur, and what they have seen is that fluid, the bone, it had malignant bone cancer, osteosarcoma. So you can imagine how old the disease is. And one of the central region of our pelvic guard. So here, when you do the computer tomography, so you can see spots, this kind of these whitish spots. So these are the symbols or these are the cases of metastatic prostate cancer. Prostate cancer, when it metastasis, when it comes out of the origin, it has a tendency to metastasize to bones. So that is why with the advanced, very important paper that actually describe what are the characteristics, what are the hallmarks of cancer. So how you will tell, okay, this cell has cancer, this cell is cancerous, this cell is transformed from its normal therapy. So there are several factors we can assess, starting from sustaining proliferative signal. Okay? You stop feeding the stop nutrition, you stop all the growth factors. Still, you will see cancer cell and the ability to proliferate. Indefinitely. So when we think about cancer, this is the first thing that comes into our mind. And cancer has the ability to so we have the capability to remove or suppress growth suppressors. If you give the cancer cell any treatment that is supposed to suppress the growth, they will eventually evade it. They have the ability to invade and metastasize. That means they undergo EMT, I think, epithelial mesenchymal transition, right? So suppose someone has tumor in prostate. The tumor cells, they will come out of the prostate gland and they will go into the bloodstream and they will travel. Eventually, somehow they will go into the bone. So this is called invasion and metastasis. They are replicatively immortal. So they are essentially immortal. So they have the infinite replicative potential. And this is tuberculosis. That means once they go out of the community, produce they have the resist cell death or death. So these are the different hallmarks of cancer. Cancer is a disease of mutations. It's not about only a single mutation. So it's like you have someone has mutation in P53, it doesn't mean they will have cancer. It's a accumulation of mutations. So a lot of mutations eventually it leads to cancer. So that's why you see that most of the cancers they are coming at the late stage of our life. So cancer, mostly people say it's a disease of old age. Okay. So this figure is actually a very complicated figure, so I'm not going to too much detail about it. So it's actually showing the intracellular signaling pathways that actually test tuberculosis. So crosstalk. So crosstalk between different signaling pathways. So proliferation signals. So how the cell can proliferate. So how the cell become viable. So they remain viable all along their growth life. Only receptor. So how the cancer cell can go out of their origin and infect other tissue. So all these signaling pathways, they are actually interwoven. So they are cross-talking with each other. So that is why it's very difficult to treat cancer. So because, for example, you come up with some drug and inhibits proliferation site, but still other circuits are on. So that is why it's very difficult to manage cancer. So that is why we can say that it's a disease of now treatment involves chemotherapy, it involves radiation therapy, and it involves debulking. That means surgery. But prior to surgery, most of the cancer treatments are done by chemotherapy. So there are a lot of chemotherapeutic drugs like doxorubicin, like pembrolizumab, lot of chemotherapeutic drugs here. But the problem with chemotherapeutic drug is they are very much cytotoxic. They are immunosuppressive. The patient
start with cycles of chemotherapy treatment. Eventually, what you will see, they lose their hair, they have severe immunodeficiency, they are vulnerable to other infections, they are vulnerable to different acute symptoms like diarrhea, thrombosis are those things, and that is why nowadays we are coming up with a new concept that is called cancer nanotheranostics. Okay. So I just wanted to update my students and also all the audiences that we can actually dig into this cancer nanotheranostics. So what it actually does, cancer nanotheranostics includes chemotherapy, radiotherapy, along with nanomedicine. And it's actually specifically targeting the tumor microenvironment. So tumor is a very heterogeneous environment. Okay. So if you slice a tumor and analyze them, you will see the tumor microenvironment full of different cells. Index cancer cell, normal cancer cell, cancer stem cell, endothelial cell, cancer associated fibroblasts. So they all compete with each other. Which means up to so you can dig into this about cancer genomics, cancer personalized medicine. So this is cancer genomic data. So now we are in the genomics data, right? So you can and if you go cioportal.com and you can get any data. So this handy, very useful genomic resource website. They will tell you the survival. All data you can get from BCG. So BCG is very robust, very handy resource you can all use. These are all public data. Anybody can use it. So I would like to just stress upon the fact specifically that whenever you use any public data, you have to cite the reference. Okay. So nowadays, like there are a lot of public data, like people are citing the data from public domain, but they are not referring. So that is baggage. So students should be very careful about it. You cannot do it. You are not allowed. Okay. So now coming back to COVID-19 cancer. So over the world, a lot of countries were trying to beat COVID-19 pandemic and cancer. So there is a very good review paper that just came out from Clinical Reviews in Oncology Immunology, and it actually showed the global contribution to the publication and how cancer patients are actually affected by COVID-19. So you can see China has the head start. So 42% publication is coming from China, followed by Italy, United States, United Kingdom, France, and France. So people really think about how to treat, how to manage these problems during our COVID. So the first comprehensive link between cancer patients and COVID-19 actually came from year on. So this paper says cancer patients with SARS-CoV-2 infection, a nationwide analysis in China. So they did it actually in China. So I'm going to show you the key results from the paper. So what they have done, they have actually carried out systematic comparison between invasive ventilation or ICU admitted patients and invasive ventilation much more resistant to COVID-19 infection. Cancer survivors, the number of severity goes up, and patients with cancer, patients with comorbid issue, that is the cancer, it's skyrocketing. Right? Skyrocketing. But you can see here, if you compare around this one, say invasive ventilation, then plus clinical indication. That is, cancer patients, they have very advanced stage of cancer, say stage 3, stage 4. So you can see almost sevenfold increase of getting COVID infection. So this is an alarming situation. They have also did after disease onset assay. So they have started looking at the patients, different clinical aspects after the disease onset. Very less. That is the clinical. You can see the difference clearly. And they have calculated the hazard ratio is almost 4. So patients with cancer, they have 4 times more chance of getting infected by SARS-CoV. And this get worse, your cancer stage is in very advanced. So very advanced stage of cancer, they are definitely going to if you have COVID-19 infection. So this paper actually first they showed age mediated. So they group them and then they look for different signs and symptoms. So if you group them in different age, you can see percentage of getting COVID-19 infection. When you have cancer, it goes up when the patients are around 6 or 8 years old. So they become much more vulnerable. When they looked at different clinical symptoms, they saw fever